Hi, everybody. Thanks for having me. So I wanted to just provide a little bit of background about what has been engineered into these trees. The SUNY ESF group has tried a lot of different things, including testing of 6 transgenes, so from unrelated species, and 26 cisgenes, which would be from related Chinese chestnuts. And they've also experimented with various markers and promoters. But they've determined to focus on a sequence that's derived from wheat, and it is a sequence that codes for the production of oxalate oxidase, which is an enzyme that inhibits the spread of the fungus on the tree. So it doesn't actually kill the fungus, but it makes it harder for it to spread enough to be lethal to the tree. Next slide, please. The question is this going to work? It's being put forth to restore chestnuts to their natural environment. So the SUNY ESF group and TACF have put a lot of messaging out there, including Buster Blight and Charlie Chestnut, and the Buster Blight is attacking Charlie, and Charlie puts up his shield, and the chestnuts are protected forever after from the blight. Which is very misleading. The tests so far on the oxalate oxidase or OXO tree, engineered tree, have only been done on very young trees and only under controlled conditions. We know that when trees go out into the forest, they could live for a couple of hundred years and face a lot of different kinds of environmental conditions that could affect how that trait is expressed. We also know that blight resistance is a very complex trait in the Asian chestnuts, which are naturally blight resistant, and that it's probably going to require multiple pathways for a long-term stable resistance to the blight. And even the SUNY ESF scientist, Mr. Powell, says this very explicitly. He says, eventually we hope to fortify the American chestnut with many different genes that confer resistance in distinct ways. Then, even if the fungus evolves new weapons against one of the engineered defenses, the trees will not be helpless. And that's because we know that engineering fungal resistance is a very tricky business, because and it's been tried in many common crops. But the problem is that pathogens like fungi are good at finding their way around plant defenses, and also when they invest their resources into defending against pathogens, it affects their growth patterns. They don't grow as vigorously, and they can be more susceptible to other pathogens. And so this is why really commercially available, there's only one fungal pathogen resistant crop that's available, and that's a potato. So, and the other issue is that even if resistance to the blight is reasonably effective, there is another pathogen, Phytophthora, or also called root rot or ink disease, which is also lethal to American chestnut and had already been the chestnuts before the blight came, and Phytophthora is spreading. Next slide, please. So it seems very unlikely that simply encoding this wheat gene to oxalate oxidase is going to enable the restoration of American chestnut into our forests. So the question is, why is this being rushed into the regulatory review? Because it's a test case, and it's being very explicitly referred to as a test case, a test of both the regulatory processes for genetically engineered forest trees, and also a test for winning over public opinion, which has generally been very opposed to the introduction of genetically engineered trees. So there are, in our white paper, many quotes and from different sources referring to how this would be a test case. And just as one example, the Forest Health Initiative, which is a major funder of the effort, says this initiative will focus on a test species, an icon of the eastern U.S. forests, the American chestnut. The American Chestnut Foundation also says if it's successful in obtaining regulatory approval for the transgenic, blight-resistant American chestnut trees, that would pave the way for broader use of transgenic trees in the landscape. Next slide, please. So what are the other trees that are being researched in the world of forest tree biotechnology? That's the broader context that we need to look at. And that is mostly focused on commercial applications, especially poplar, eucalyptus, and pine species. And it's mostly about commercial uses, producing more biomass, withstanding the stresses of plantation forestry practices, altering wood characteristics, and especially modifying the characteristics of lignins so that wood can be more suitable to the production of biofuels. And researchers such as Steve Strauss, who has been very prominent, they're very explicit about this. We use modern plant biotechnology to help create environmentally sustainable biotechnologies to aid in the production of crops, renewable energy, wood, paper, ornamentals, and fruit. So it's not focused on forest health for the public good. Next slide, please. What are the risks of introducing GE American chestnut or any other GE tree? It's contamination of other trees of the same species, ecological impacts to the water, to the soil, species. Also, in the case of chestnuts, human health, because people do eat chestnuts and inhale the pollen. And the question is, can we really assess the risks given the complexity of forest ecosystems and given how little we know about them and how little we really know about the American chestnut's role in those ecosystems? Next slide, please. So the even broader context is that biotechnology is really expanded recently with the development of a lot of new technologies. Gene drives, gene editing, de-extinction, designer babies, engineered forests. The question is, where do we draw the line? Decisions that we make now will be precedent setting for the future of biotechnology regulation in general and for the application of biotechnology to forests and to conservation. Thank you for having me.